thank you very much to the organisers, particularly Simon Hawke, who's done a great job putting today together. And like others before me, I just want to say congratulations to Simon Hammond for really an amazing career. And I think the quality of the speakers are, of course, present company accepted, really speaks to the work that you've done over the years. So again, I'll say mazel tov. All right. I'm allowed to say that because, of course, Jamie Gordon reminded me today that he saved me for the second time in 20 years from eating a ham sandwich at lunch. So today I've been asked to talk to you about advanced imaging, and you'll see one of our MS neurologists, Todd Hardy, who was recently described in the Sydney Morning Herald as, I can't remember, something like a russet-haired goatee protruding from his face, which you can see just here, performing some advanced imaging techniques in our clinic. And our clinic really is the crux of our research, as well as our clinical practice, of course. And it's important to emphasise that the crux of our clinic is our patients, and they really drive the research. And this is just a little geodata distribution of our MS clinic at the Brain and Mind Centre. And for those of you who have keen eyes, you'll see a little dot up here which represent the two patients that Jamie Gordon has referred to me with MS in relatively recent times who've come down to our clinic. So we appreciate that. And this slide is just to say that imaging is but one facet of the research we're all doing in MS, but more and more we're trying to tie these elements of our research together with the help of our current and past fellows, and there's some suspicious looking characters, one of whom is in the room today down the bottom. So imaging in MS. So I'm really going to give you a bit of a potpourri sort of tour. I'm not going to give you an exhaustive list of imaging techniques in MS. We all know that this is the typical picture. This was actually a 23-year-old man who came to see me. It must have been 2004 because we were in the old building at the Brain and Mind Centre at the time. And he actually had primary progressive MS, and unfortunately he's now very disabled. But his MRI at presentation, you can see these typical pericallosal lesions. Dr. Masters, I know you're still in the audience. You told me you were leaving for my talk, so excuse me if I get any of this wrong. So pericallosal lesions on this flare sequence, and here he had these significant brainstem lesions, which was no doubt driving his progressive gait ataxia. And sometimes we see on newer sequences like this susceptibility weighted scan the association of MS lesions with a central vein, and you can see a nice central vein coursing through the middle of this lesion. And that's quite interesting because some of the more recently described diseases such as neuromyelitis optica don't tend to have that central vein, and we can distinguish the two entities at least 70 MRI on that basis, according to some researchers. And here we see typical spinal imaging. This is in a patient with very aggressive relapsing disease. In fact, this is Jane Frith's patient. Sorry, Jane, for not asking permission to show this, but and you can see this typical eccentric lesion here. So we've all seen these patients, and some people today have already spoken of the correlates with MS pathology, but this is just to remind you. This is another one of John Prenius's lovely slides at the top left here showing an acute MS lesion stained for myelin oligodendrocyte glycoprotein. And you can see these round structures are all macrophages that have infiltrated the tissue. I was interested, Martab, in your talk before when you talked about resident microglia. Interestingly, in most acute MS lesions, you don't see many resident microglia, and that's probably because many of them have actually transformed into macrophages. So some of those macrophages are locally derived, and some of them come, of course, from the blood. And if you can get your eye into this picture, you can see some longitudinal profiles which are axons that still have remnants of myelin on them. And this very typical active phagocytic picture correlates, as John Pollard said earlier today, with the MRI evidence of gadolinium enhancement. And in this scan, you can see multiple gadolinium enhancing lesions. And this, in contradistinction, is a chronic, punched out, typical MS plaque. Unfortunately, our standard imaging sequences, for the most part, don't distinguish what is happening at a pathological level, and certainly not at a pathophysiological level in these MS lesions. So a T2 lesion, which you're all used to seeing, could be a demyelinated plaque, could be a remyelinated plaque, might have varying degrees of inflammation, gliosis, axonal loss. It's quite hard to tell, at least on a T2 scan, although there are some aspects which we can tell, including the degree of severity in axonal loss if we combine that with other sequences, such as a simple T1 scan, and that's illustrated here. Here we have an acute MS lesion stained with non-phosphorylated neurofilament. This is Bruce Trapp's work from 1999 showing these beautiful axonal spheroids. And of course, this was really a recapitulation of the German literature from about 150 years earlier, showing exactly the same thing. And we know that that does correlate on imaging with the development of black holes, which are areas of significant axonal destruction, and we know that they correlate in turn with clinical outcomes far better than just standard T2 lesions. Martab has already referred to these subpial lesions. This is from Claudia Lucanetti's work showing this typical subpial lesion, and we know, of course, that the deep grey matter is also terribly involved in MS, and this is one of John Prunius's whole brain coronal Luxol-fast blue-stained sections, and you can see here's the thalamus, here's one lesion, here's another lesion. This hippocampus is totally demyelinated, caudate nucleus is demyelinated, and in addition to more typical deep white matter plaques, and here's a lesion in the corpus callosum. So in one section already multiple deep grey matter lesions, and we know that that drives many of the clinical outcomes in MS, particularly cognitive outcomes. But unfortunately imaging really doesn't show the cortical lesions, at least very well. This is a patient I saw in my days at St George Hospital who came in to hospital with paroxysmal stuttering as her first manifestation of MS, which I have never seen since and don't think I will, and that paroxysmal stuttering was in fact due to seizures, and she had a very high burden at least of subcortical disease, and probably, based on looking at a standard flare sequence, cortical lesions. And in fact, I remember sitting there with Mark Hirsch giving this lady some midazolam intravenously, and her seizures stopped and she started speaking normally. And she actually has gone on to do very well, although she had a period of psychosis for some months after that, no doubt also related to the burden of her cortical disease. So MS can present in strange ways. Newer imaging techniques do help us a bit in this regard, in that at very high field imaging, 7T, 8T, we can see cortical lesions much better. And even when you compare 1.5T to 5T in this case, imaging, you can see numerous lesions that you would otherwise miss on a 1.5T scan. So probably when we scan our patients and we say, you've only got a mild burden of disease, we are probably only seeing the tip of the iceberg. And there have been developments in a number of techniques to help visualize these beyond just increasing the field strength of the magnet. This is double inversion recovery, a sequence that suppresses the signal from both CSF and white matter. And you can see here juxtacortical and cortical, true cortical lesions in these scans. And if you see this, it's a significant prognostic marker, and certainly if you see it in a patient with a clinically isolated syndrome, they've got a much higher chance of converting to clinically definite MS than just the presence of a deep white matter lesion. The problem is that even with this technique, we're probably only seeing 20 to 25% of the cortical pathology that you can discern under a microscope. So look, that's the background that you probably all already knew. More importantly, in the modern era, with the advent of modern therapeutics, is the capacity to track our patients longitudinally and see what happens with time. And I think MRI has had a profound impact in this regard. From the very simple things like the one you see here, we all look at our scans. Our patients come in and we say, look, have you got a new lesion, have you not? And Lynette Masters spends probably 2/3 of her life doing this, just sorry for that sad sort of reflection on your life, Lynette, but I think that's probably a fair comment that I go down there and Lynette is literally looking from side to side at these MRI scans, which now come with about 300 slices. These are not big, thick slices anymore, and it takes a lot of time. So there are simple things that we have been doing in clinical trials for a while and now are coming to the fore in clinical radiology practice, such as subtraction imaging, to allow us to rapidly see new lesions, but also shrinking or disappearing lesions to help monitor our patient's disease. And you can imagine that if a patient comes in with a scan like this, where there are 667 regions of interest comprising 243 lesions, seeing one or two new lesions in amongst all of that is actually a painstaking and usually impossible task. And this is just that same patient with those lesions represented in 3 dimensions. So that's lesions. What about brain volumes? This is a slide that Alex Rivera kindly gave me, just showing a patient really, in essence, before the treatment era, or at least before the modern treatment era, showing progressive brain volume loss, and you can see that with the expanding lateral ventricles. Obviously, there's an accumulation of T2 lesions, and more importantly, an accumulation of black holes and enlargement of those black holes over time. And unfortunately, by the time the patient gets out here, and in this case, this patient was in their late 30s by the time they got here, it's all a bit late. This patient's probably already going to have to leave the workforce soon if they haven't already, and will almost certainly have significant physical disability as well. And our treatment goals are really defining the way we monitor our patients over time so we can prevent them getting to that stage. And this concept has already been raised, this concept of NEDA. We used to look at relapses and disability progression, but now we are really using MRI very intensively in our clinical practice to ensure our patients don't have disease activity in the form of traditional lesion metrics, new lesions, gadolinium-enhancing lesions, and now not yet made it to clinical practice for a variety of reasons, but we are now looking at least at the group level at brain atrophy, and we know that patients whose brains shrink at more than 0.4% per year are probably not optimally controlled. Our brains, at least most of us in this room, are shrinking at least about 0.2% per year, probably a little bit more. That's normal. So how do we implement this in clinical practice when we want to look at lesion metrics and we want to look at volumes? I think we're now getting to the point that we're beyond the stage of just having descriptive qualitative reports. Sorry, Lynette. Lynette and I have had this discussion many times, but I think we will start to get to a point where there is a demand very soon for quantitative metrics in our reporting. Helmut Butzkerven once said that if you go to your bank and you ask how much is in your account and they say it's pretty much the same as last time, that's simply not good enough. We actually really do want to know what has changed at a quantitative level. And I think these sort of reports are coming. This is from our group, but there are now commercial groups who, for $50 to $80, will provide you with a quantitative report on an individual patient. Now, there are a lot of caveats to that, and there are a lot of difficulties in the interpretation, particularly of brain atrophy at the individual level. But as those problems are solved, I think we will start using this. How else can we use quantitative data of brain volumes right now? Well, this is just some interesting work I'm going to take you through from our group. This is from Tim Wang, who is a PhD student in our group, who's looked at, can we use a single clinical scan in a patient who comes along to see us to predict how that patient's going to do in years to come? And it's quite interesting that you actually can do that and get it right almost or most of the time. And you can actually provide a clinician with a probability that a patient's going to do badly simply by measuring their brain volume at a particular time point. And that's based on comparison to large data sets of normative data, i.e. normal healthy controls, who are age and sex matched. And if you can say to a patient who's 30, well, your X standard deviations away from normal, and you know when their disease roughly started, and I mean roughly because we all know that symptom duration is not really an accurate reflection of when this disease started. It probably starts well before that. Then you can say, well, if you project backwards to the time you had your symptom onset, and we assume, and it is an assumption, that you had a normal sized brain then, we can actually determine the rate at which you have lost brain volume up until the current point, and we can probably, given that brain atrophy is relatively linear in MS, see how you're going to do over the next few years. So we validated this work using clinical trial data that Novartis has kindly provided us, looking at the freedoms data, particularly in the patients who were placebo controlled, and we can show that the patients who had small brains relative to what they should have been at that age, they actually do very badly independent of whether or not they have disease activity by conventional metrics in the first year. So whether they get new lesions, gadolinium enhancing lesions. So this is just a simple way that we might be able to use brain volumes to predict how our patients are going to do and integrate that into clinical practice. But I think the future is more in looking at large data sets of patients with this disease, with MS, and we're sort of starting to make some headway into this by looking at data sets from a number of groups around the world and merging them. And I think the future will be in the application of techniques such as artificial intelligence to these data sets to find things in our images we are not even aware are there. So we probably look at our scans, we're looking at the volumes, we're looking at the lesions, but there may be a whole lot of other things that we don't even recognise. And it's techniques that are sort of unbiased, like artificial intelligence, which I think will take us there, not just in terms of diagnosis, but more importantly in terms of disease monitoring and monitoring of treatment response. This slide I borrowed from Heidi. I just threw it in there to say there are an enormous number of advanced MRI techniques that I am simply not going to mention today. But I will now move on a little bit to just mention some of the other work that I think is of relevance. I've talked about brain atrophy and lesions. It is important to recognise that they affect each other. So many longitudinal MS studies, particularly those in which we apply these advanced imaging techniques I listed in that slide before, such as DTI, MTR, to try and explore what is really going on at the pathological level in these lesions, have been unfortunately applied inappropriately in many of the studies published. And that is because longitudinally the brain shrinks and the lesions move, and none of these studies have taken this into account. So you can see here, if you draw your lesions at baseline and then you follow the patient up and you just project those lesions onto the follow-up scan and then see what's happened in those lesions, unfortunately the lesions move, and often they move into the CSF, for instance, those that abut the ventricles. So you can imagine if you're trying to look at sensitive techniques like DTI and suddenly half your lesions in the CSF, it's a completely confounded way of looking at these images. So we have now developed techniques. These are about to be presented in April at this conference, which is an imaging conference, to be able to actually use a technique known as localized lesion mapping to move these lesions into the appropriate place, so we can actually say what is happening within that lesion at follow-up. And I think this is going to be very important when we come to look at the pro-remyelinating therapies that Koinianikis has already alluded to. And not only will we be able to look at what's happening in the lesion, this technique seems to be so precise that we can look at what's happening in the lesion core, what's happening in the lesion rim, what's happening in the periplaque white matter with great accuracy. And it may turn out, for instance, that remyelinating drugs have a profound effect at the lesion rim, but they're simply in the lesion core. You've just lost so many axons, you just can't remyelinate. There's nothing there to remyelinate. And some of the work that's come from Sasha Klistorner quite recently has shown that in the core of a lesion, the diffusivity, the mean diffusivity, progressively increases in chronic disease. And that probably, it's not surprising, it probably reflects that you've got progressive axonal dropout in these lesions in which the surviving axons are not remyelinated, and there is progressive attrition of those axons over time, which might underpin the progression in this disease we see clinically. This slide is similar to the one I think that Con showed before as well. So we can now apply these sorts of techniques. This is a paper on diffusivity in the optic radiation in MS. The optic radiation provides very nice fodder for the study of lesion evolution in MS, because the optic radiation is a coherent fibre tract, all the fibres running in one direction, which takes away the confounding effect on advanced imaging techniques like DTI. So for instance, elsewhere in the brain where there are crossing fibres, it is very hard to interpret what is happening in one fibre tract or another. One of the things that's quite exciting that we're doing is, and again, Con Yannicka stole my thunder here, is we are not just looking at imaging in isolation. We are now looking at biomarkers in which we combine imaging with electrophysiology. In this case, with multifocal VEP, and previously we've done that in the vestibular system with electrical vestibular stimulation. And the combination of these techniques seems to be even more powerful. So here, of course, is the multifocal VEP being acquired. And we are now looking at lesions defined structurally on MRI and functionally with VEP. We are even able, and this is Tim Wang's work again, to come up with algorithms that allow us to, for the very first time, actually measure the conduction velocity in the central nervous system in demyelinated lesions. And I think Con has shown this in the optic nerve, and we've shown it previously in the medial longitudinal fasciculus, but here we've shown it now in the optic radiation. And it's quite interesting when you look at this figure. The lesion conduction velocity sits around 2 meters per second, and it's quite interesting if you go back to some older literature and hear from Ken Smith, where he said no one's really been able to do it, but he predicts that the conduction velocity is likely to be no more than 1 to 3 meters per second in demyelinated CNS. So it was a pleasing result to show concordance with Ken Smith, because it's more likely to be believed. How are we going to take this further? Well, I think that the next step will be, as I say, to apply these techniques that we are developing to much larger data sets, not just lesion metrics, not just atrophy, but the sort of things I've just shown you now. And one of those ways is collaboration, and this study that's coming up, which is it's known as the 5-centre study, but since Helmut has moved hospitals, it's now a 6-centre study. And we are going to be doing the largest longitudinal cohort study looking for imaging biomarkers of subclinical disease progression in MS ever done in the world. All of this imaging is going to come back to the Brain and Mind Centre for analysis. We hope not just with conventional metrics, but with our other paradigms, to see if we can find markers that we can then translate back to the clinic. An even better way of doing that is to take a much larger cohort, not just 5 centres in Australia, but maybe we can do this globally. And there is now a proposal on the table which is being driven by the Brain and Mind Centre and specifically by Heidi Beadnell to integrate an MRI repository into MSBase. So Karen's already talked about MSBase having 50,000 patients in it, fantastic clinical data set, but it's totally devoid of real MRI data. The only MRI data in there is just what a neurologist says, is there 9 lesions, is there not? It's pretty hopeless. So one of the criticisms that comes back from Brain and all these other papers that Thomas is publishing vigorously in is, well, that's great, but there's no MRI data. This is an MS paper. How can you possibly have an MS paper with no MRI data? So Sydney University, in collaboration with MSBase, hope, and I say hope because it's not formalised, this agreement, to start building a repository whereby centres who participate in MSBase can upload their scans. Those scans are de-identified, and just like the clinical data belong to the person who sent them, the PI at each site, and that data will sit there, and if one of you who want to do a study and you are an MS-based PI, you will be able to access not just the global clinical cohort, but the global MRI cohort. And sure, there will be different acquisitions, different machines, different levels of quality, but if we really get thousands and thousands and thousands of patients in it, I think we will overcome that to a great extent. Lastly, I'll just put in a plug for the MS Research Australia Brain Bank. As you know, that is run from the Brain and Mind Centre, and I inherited that from Simon Hawke. Thank you very much, Simon. And then I devolved part of it to Michael Buckland, who's the neuropathologist at Royal Prince Alfred Hospital. For the last 3 years, we actually have been acquiring MRI scans on all of the brains that are donated as they come through the Brain and Mind Centre's scanner, and Dr Masters has kindly been acquiring these images for us. And we are yet to interrogate this in any depth, but what it has done, at least at a simple level so far, is allowed us to co-register the images with the pathological slices. And when we do that, we are then able to localise the lesions and tell our pathologists where to go back to look for very small lesions or specific types of lesions that they may not have seen. You've got to remember, there's no way we can block out every block in the brain and do 1,000 stains on every block, because that would be impossible from a manpower perspective. But if there are specific lesions that are of interest, or a research group around the world has a specific interest, we can pull out all of those that have lesions in a particular area and go back to the pathologists and tell them where to sample. And a great example of that is, in fact, an American group who's requested samples of patients who have lesions in the thalami. So we've now been able to go back and pull all those out based on the MR imaging. And this is an example. It's almost impossible. You would never see it with the naked eye. It's quite hard to see in this light on the imaging, but trust me, it is there. Little lesion, and we went back and looked at it, and in fact it was a remyelinating lesion there. So again, great to have this capacity, and we will interrogate these images further with newer techniques, but that's for another year. On that note, I think I'll finish, and happy to take any questions. Thank you.
Any questions for Michael? Con.
Michael, I was surprised with your brain atrophy projection to be able to predict outcomes some way down the track.
Yeah, so were we.
Why it didn't relate in any way to relapses or the number of handsome lesions?
Yeah, look, I think that was very interesting. What I didn't tell you also is that, not from our prediction, but if you look at the patients who have small brain volumes relative to healthy controls at the beginning of the Freedom study, and this is about to be published, that sure, a lot of those patients got treated over the 8 years of follow-up data available so far, but in fact you can determine who is going to do badly 8 years later based only on the brain volume, and you specifically can tell, even if you by looking at patients who only had brain volume loss in the first 2 years, who's going to do badly at 8 years, independent of the presence of relapses or the presence of new lesions forming. Why is that the case? Is that your question? Yeah. So why is that the case? Look, I don't know. I personally don't think these are totally independent processes, if that's what you're getting at. I think that they are interrelated, and it may be that the damage was already done from a lesion perspective prior to that, and we'd caught it too late to be able to show that.
Because the other component of that question is the linear analysis of brain volume. If you don't have linear sort of onset of relapses or linear sort of new lesions,
Yeah.
so it would suggest that the process occurred earlier.
The process occurred earlier. Yeah. And it makes sense that early process is the inflammatory process and the lesion formation at that stage.
So,
If I would just make a comment, I think that the atrophy data found with the Bavaggio studies have sort of demonstrated the converse of that, so that if you don't show atrophy occurring, those people many years later do much better.
Yeah.
So.
Yeah, that's true, and in fact that's absolutely right, and that came out of the Freedom study we looked at as well. Same thing.
So are the number of lesions you have at the point that you measure the atrophy relevant rather than stimulation?
You know there's this paradox between lesions and brain atrophy, and I think the explanation for that in part is that brain volume is not brain tissue. So brain volume is comprised of tissue that we are hoping is there, axons, neurons, glia, but it also comprises inflammatory cells, gliosis, and water. And how you account for those in our measurement of brain atrophy to get a measurement of what we like to call brain tissue loss as opposed to brain volume loss is hard. We are working on that, and we have managed, and I think you are involved in this research, Con, to sort of come up with at least a mathematical model that allows us to, in essence, eliminate water from our measurement, or at least to correct the atrophy measurement for water content. But how on earth you correct for inflammatory cell content, gliosis, etc., I think we're a long way from that.
Okay.
Jeff.
Oh,
If there's time, just a quick question. Right, do you have any work yourself on AI here, or is that something that you're aware of that's happening around the world?
Yeah.
It seems to lend itself to it if you can develop metrics,
Yep.
get a deep, neuronal system processed over deep learning.
Yeah.
It's just, if they could play Go better than a human being has ever played in two millennia,
Yeah.
it sounds like that's right up.
That-
Is people working on it?
Thank you for asking that question. I can see Patsy smiling because we've been in deep discussion about this, and now we're going to build this. And in fact, Sydney University does have very substantive deep learning and machine learning expertise, particularly since the appointment of a world superstar in that field, whose name is Ducheng Tao, who I think is probably one of the top 1% cited authors in the world in this field. And he is joining a group of us to try and build this. It's already happening to some level in other fields, and it's happening commercially. So there's a couple of entities around the world now where for $1, so you can look this up, zebramedical.com. For $1 you can upload an image, so far not a brain image, but virtually most other organs, CT, plain X-ray, MRI, and you will get back an AI-driven report from the website. Now, what the quality of that is, I have no idea at the moment, but undoubtedly it will get better and better.
we better keep moving along.
Thank you.
Sorry. Oh no, Simon wants to talk. I thought he was trying to move us.
How soon will it be before we're going to be using volumetric data to make clinical decisions about treatment?
Simon has already told me he doesn't believe in any of this gobbledygook. I think that will probably only come when we have therapy that we know specifically targets that. And there are nuances between current therapies in terms of the effect on brain atrophy, but that probably reflects what Con says, and that is its impact on inflammation rather than on the true processes that drive neurodegeneration. But once we have neuroprotective therapies and once we have pro-remyelinating therapies, then I think it will become much more important. But if you said technically when can we do it, you can do that and get real-time reports from a commercial vendor now. It's what does it mean is the real question. And I don't think we're there yet.
And the other point is talking about your predictive model. I mean, should everyone who gets an MRI in an MS centre or even in Orange, for instance,
Even in Orange.
we have a T2, we have two T2, sorry,
T3, yeah, 3T.
3T magnets.
Yes.
And I mean, just need to do
there will
T1
soon,
slides.
I wasn't really going to say this, but there will soon be a website called brainatrophy.com.au from our group, which you can drop a whole DICOM file on, and it will run this predictive algorithm and spit back and tell you what's going to happen to your patient over the next few years. Now, a lot of caveats to that. Research only, not for medical use.
Does that include like drugs, steroids? I mean,
Yeah.
how are you going
That's
to-
the problem. This is, at the moment, that data has only been validated in an untreated cohort. So absolutely, Lynette's right. If you have steroids, you can shrink your brain by up to 2% within the next couple of weeks. If you're dehydrated, these are all the biological confounders, disease-modifying therapies, etc. And none of that has been put into the model at this point. That is why there will be a series of disclaimers. It is at the moment just an interesting research.
Don't forget all that fine wine that's out here in Orange.
I'm thinking of it.
Yep.
Thank you, Simon, for this nice invitation to this great meeting. It will be my pleasure to share with you some data we generated over the last few years with Simon in the field of microvesicles, which are now the new names for microparticles. So in my slides you will see the word microparticles quite often. We have been working in this field since, we can say, since the end of last century, and the interest in microparticles came from our studies in inflammatory diseases of the brain. And well, the title here that Simon suggested to me, biomarkers or something more, is obviously a title in which I suggest that there are other possible roles for microparticles or microvesicles. We would like to propose to you that microparticles are active players in inflammatory disorders, and particularly in MS. So my lab is interested in endothelial cells. Oh, this one doesn't work. Endothelial cells, particularly those of brain vessels, blood-brain barrier in particular. We've been able to characterize the few changes occurring at the level of brain endothelial cells, and notably the production of microparticles. So I'd like to briefly introduce to you what microparticles are, or microvesicles as we call them now, and then I'll be happy to share with you what we know now in terms of plasma levels of microparticles in MS patients, and I'd like to talk about some new possible ways to characterize these vesicles. So first of all, microvesicles or microparticles are not something completely new. They were described by Peter Wolf in '67. They were called platelet dust, and I remember in '78 I had a patient with vasculitis, and when we made a biopsy it came back with this diagnosis of platelet dust, and platelet dust were in fact platelet microparticles that were produced in these lesions. Then it remained relatively silent, and in the 90s microparticles were studied by experts in platelets and coagulation, and it's only near the end of last century that we made the link between microparticle production and inflammation. We showed that TNF, tumor necrosis factor, can increase the production of microparticles. And microparticles are produced exclusively at the level of the plasma membrane, while other extracellular vesicles, such as exosomes, are produced inside the cells and have very different properties. There are also apoptotic bodies, but we don't study these at the moment. So we showed that when you stimulate endothelial cells, and notably brain endothelial cells with TNF, you markedly enhance the production of those little blebs at the level of the plasma membrane in a dose-dependent manner, and you can block that with anti-TNF antibodies. So we started to analyze the possible role of these microparticles in inflammatory pathologies. Well, the way they are produced is something entirely physiological. They are constantly produced by the flip-flop mechanism in the plasma membrane. That means the exchange of the lipids between the inner leaflet and the outer leaflet of the plasma membrane. There are 3 enzymes which control that kind of movement: the flopase, the translocase, and the scramblase. And this results in the production at the surface of the cell, the production of those microvesicles or microparticles. And if you look at the cell surface from the top, you will see those kind of protuberances which precede the release of these microparticles, either in the culture supernatant or in the circulating blood. And I'll come back to this kind of protuberances at the end, when we will see that you can modulate the production of microparticles by fingolimod, notably an important drug in MS. So our hint into the role of microparticles in pathology came from our studies in cerebral malaria, another disease that affects the brain. We found out that there are at least 2 levels of complexity in cerebral malaria. Well, first of all, obviously, the interactions between cells that are in the vessels and the brain endothelium itself. But the second level of complexity is that each cell type here, the red blood cells, the platelets, the monocytes, and the endothelial cells, can produce these cell-derived microparticles. Microparticles, as I said, are produced constantly in physiological conditions. So in our plasma, we have billions of microparticles that are in circulation. It was found that some agents can enhance the production of these microparticles by profoundly altering the biophysical properties of the plasma membrane. very complex changes in membrane lipid asymmetry. And this stimulation, notably by TNF, can result in the increase in the number of microparticles. So microparticles will be produced threefold or fourfold in higher numbers. And each microparticle is defined as a submicron element that carries, of course, antigens from the cell of origin, but also molecules such as phosphatidylserine, which is normally hidden inside the membrane, and in the case of production of those microparticles is expressed outside. Microparticles carry a phenotype that is identical to that of the mother cell. By mother cell we mean the cell type that has produced the microparticles. Indeed, here you see there is a lot more ICAM1 on brain endothelial cells when you stimulate them with TNF, while the same is true for endothelial microparticles. That's how they look like in scanning electron microscopy. Here you see activated platelets, and here are the microparticles, those spherical elements. There are other vesicles, as I said before, notably exosomes, which are at the frontier between 100 or 200 nanometers, but by and large, microparticles are between 200 and 500 nanometers in diameter, so they are in the nanosize range of elements. To summarize what we did in the field of cerebral malaria is that, well, we have high levels of these particles at the time of the neurological syndrome, both in an experimental model and in patients. When we block the production of microparticles, we block cerebral pathology, and conversely, if we take microparticles from mice with cerebral malaria and inject them intravenously, we can trigger pathology. So these are the arguments suggesting that they have a role really in pathogenesis. Together with Bernie Saunders and Warwick Britton, we have also studied what they can do in the field of TB, and in that context, we studied the effects of these microparticles on target cells, notably on antigen-presenting cells and on T cells. We were able to show that microparticles become compartmentalized in macrophages, taken here as a target cell, notably found in the recycling endosome here. And furthermore, after this kind of internalization, we showed that microparticles can actively stimulate T cell proliferation, and even more interestingly, the antigen-specific, the peptide-specific T cell proliferation is greatly enhanced by these microparticles. So this means that we have small elements of the plasma membrane that can have an important role in pathology. In the case of cerebral malaria, we show that they play an important role in sequestration and in the increase in procoagulant activity. In the field of sepsis or septic shock, we show that these particles do change the transendothelial electrical resistance, notably. In the case of TB or some viral encephalitis with Nick King in Sydney also, we show that these particles play a role in terms of spreading of the antigens, and in the field of cancer, we show that these microparticles can retemplate other cancer cells. So we asked, knowing all that, what could be the levels of microparticles in patients with MS, and what could be details of their phenotype and their contents. Well, the first question was addressed by simply analyzing by flow cytometry the numbers of circulating microparticles. So here they are shown in between platelets and the electric noise of the flow cytometer. And we found that total microparticles, that means microparticles that are picked up by the presence of phosphatidylserine, is significantly increased in MS patients, Simon's patients, who were sampled before treatment. And interestingly, those levels of total microparticles, that means microparticles coming from any cell type, went back to normal in those patients who were treated with fingolimod. This was also true for endothelial-derived microparticles. You can see here that there was a significant increase in untreated patients, and these levels went back to normal upon treatment. Now, we analyzed, of course, every cellular origin of these microparticles in MS patients, and we found some interesting changes here, which are not so clear with this kind of diagram, notably about T cells, but when we plotted the exact levels of T cell-derived, monocyte-derived, or platelet-derived, we could see, intriguingly actually, no difference in terms of T cell-derived microparticles or monocyte-derived microparticles. Interestingly, higher levels of platelet-derived microparticles, but for the first time, in 18 years that we look at microparticles, we saw a decrease in the levels of B-cell derived microparticles in patients with MS before they got treatment. And interestingly also, these lower levels of microparticles from B-cell origin went back to normal upon treatment with fingolimod. And that's where, of course, we saw that these could reflect the production of particles by regulatory B cells, B-regs as they are called now, and that's why we started to collaborate with Scott and Felix, who is the next speaker, the next two speakers, trying to address at the moment what is the role of B-regs, of regulatory B cells, in the production of these particles. And indeed, if we compare what is happening with endothelial microparticles here on the left and B-cell derived microparticles here with fingolimod, we can see that, interestingly, most patients have higher levels of these B-cell derived microparticles, suggesting a role of B-regs again. Well, just a word about the MP content, because before we started to work together, Simon had shown that there were interesting changes in the brain of patients with MS. Microparticles are not just a bleb of the plasma membrane, they also take inside this bleb of the lipids some cytosol, of course, of the mother cell, and inside there are lots of nucleic acids, mRNAs and microRNAs, which we have studied so far in the field of cancer, essentially. But we asked the question about what is happening in microparticles produced by MS patients. So that's the work of Simon here, who showed that in the normal appearing white matter, a number of genes were upregulated, or at least dysregulated, and one of them is osteopontin here. Osteopontin is one of the gene products that we targeted in microparticles, microvesicles from patients, and indeed we were able to show that osteopontin, also called SPP1 here, is significantly elevated in patients with MS and untreated MS, and these levels go down upon fingolimod treatment. So what about the effects of the microparticles produced in the context of MS on some target cells? Well, as target cell, we took brain endothelial cells, of course. We labeled the microparticles with a lipid staining agent, and when we add these microparticles to brain endothelial cells, we can see that they are indeed binding to the surface and getting internalized in these endothelial cells. And as a result, microparticles, which bind only in some areas of brain endothelial surfaces, these microparticles trigger an upregulation of ICAM1, CD54, and also of VCAM1, CD106 here. So these microparticles produced in the context of MS do have a pro-inflammatory effect. And finally, just to say that we were intrigued by the levels of microparticles upon fingolimod treatment, even though we have other modulators of microparticle production, we asked the question whether fingolimod itself could modify the endothelial level of activation in general, and more specifically, could modify the release of microparticles. We know lots of things about fingolimod mechanisms of action, and notably all the mechanisms that are related to the modulation of sphingosine-1 phosphate, of course, but we thought that perhaps on brain endothelial cells, fingolimod could have an effect. And intriguingly, when we looked at the surface of endothelial cells that had been treated in vitro exclusively with fingolimod, remember the picture I showed you initially with the protuberances on the surface of endothelial cells from the brain? Well, those protuberances that are increased by the stimulation of endothelial cell by TNF were nearly completely abrogated by fingolimod treatment in vitro of brain endothelial cells. And when we quantified this effect, not only by scanning electron microscopy, but also by looking at what was released in the supernatant, we found that indeed, at, let's say, regular doses, fingolimod was able to reduce the production of TNF-induced microparticles with a, let's say, a fair dose already here, 10 nanograms per ml, and even when we had a maximal stimulation of endothelial cells with 50 nanograms per ml of TNF, we were able to put this down with fingolimod, again, exclusively in vitro. Now, I'd like to spend the last few minutes talking about some new ways to characterize microparticles, and notably using what we call vibrational spectroscopy. We've been using Fourier transform infrared spectroscopy, Raman spectroscopy, and a few others to characterize what is happening in brain lesions. And again, it's been in the field of cerebral malaria that we were able to identify some changes by vibrational spectroscopy. Well, vibrational spectroscopy has a few advantages over classical omics approaches, as we call them, proteomics or lipidomics or transcriptomics, etc. It allows for a semi-quantitative evaluation of changes in cells at the atomic level, and we can also detect conformational changes in molecules. We were able to show in the brain of mice with cerebral malaria, again, that there are changes here around vessels which can be pictured by those vibrational spectroscopy images. And I won't enter in the detail of this, I don't want to bother you with that, but by selecting some areas of the spectrum, we can detect beta sheet protein changes. We can also detect very minute changes in the lactate content around hemorrhages. So we just asked the question, can we use those vibrational spectroscopic techniques to image microparticles? And for that, we use a very simple model of microparticle or microvesicle release upon LPS stimulation. We analyzed here those, let's say, physiological microparticles in the bottom versus the LPS-induced microparticles. And indeed, it was possible, using the second derivative of the Fourier transform infrared spectroscopy images, it was possible to detect a number of changes that would characterize physiological microparticles as opposed to inflammation-related microparticles, notably increases in phosphatidylserine, decrease in phosphatidylcholine, etc. Again, I don't want to bother you with the details, just to suggest that this technique is indeed operational on microparticles. And when you do PCA analysis, principal component analysis, from these spectral images, you can totally segregate normal microparticles versus inflammation-induced microparticles, and you can also segregate them over time. So what we are doing together with Simon now and Peter Ley in the School of Chemistry is to analyze the microparticles that are circulating in the blood of MS patients. And had this meeting been held in early 2018, we would have been able to give you the results, but we are confident that we now have another way to look at microparticles by using vibrational spectroscopy. So, to conclude, we can say that total microparticles and microparticles produced by endothelial cells and platelets are higher in patients with active disease. Conversely, B-cell-derived microparticles are reduced in healthy controls. Fingolimod regulates this to bring back those levels to normal. This is also true for the degrees of transendothelial migration, which I didn't talk about today. In terms of content of these microparticles, well, osteopontin and possibly CD47 have an important role to play, possibly in terms of biomarker, and we found out that the drug that is so efficient in MS patients can modulate directly microparticle release by endothelial cells. So we would like to propose that in addition to the cells that are in the blood and interacting with brain endothelial cells, various subsets of microparticles are being produced during MS and other inflammatory diseases, and these microparticles modify endothelial cells from the brain in order to explain changes in terms of permeability and also transendothelial migration related to these endothelial changes. So microparticles certainly are interesting biomarkers. They possibly could help us to predict when a patient is cured, for sure, but we would like also to suggest that they play an important role in the pathogenesis of MS, and we are trying to understand that better. So I'd like to thank Anna Singer, who did most of this work, also some PhD students in my lab, Pierre Julliard and Annette Julliard, of course Simon, and thank you for your attention.
Professor Pollard has a question.
Thank you.
I think my question was answered by your second last slide. I was going to ask you whether the enhanced particle production, like is related to endothelial permeability.
Yes.
Judy Spees a study of barrier permeability found that TNF was very powerful to make the barrier permeable.
Yep.
And I presume in cerebral malaria, it's a very leaky area.
Yes, actually in cerebral malaria, both in patients and in the mouse model that we study since many years now, there is I would say a mild degree of BBB leakage. It's probably more an activation of the endothelium and the sequestration of cells inside the vessel. So in the case of MS, it's not in front of such an audience that I should say that, there is a lot more transmigration than in cerebral malaria. And what we propose here is that not only the circulating cells can modify the brain endothelium, but also the microparticles that are released by these cells can disseminate a pro-inflammatory and pro-coagulant phenotype. So that's one aspect I didn't discuss much actually, but these microparticles being in circulation can propagate the pro-inflammatory potential. So they amplify, if you wish, the effect of cytokines such as TNF.
Sue?
Yeah.
How is fingolimod actually acting directly on the platelets? So have you done that in vitro? Is there any sphygosine receptor inside the platelet?
I'm sure they are, but to answer your question, we don't know yet how this can work. We haven't studied fingolimod on platelets directly yet, but only on endothelial cells, because we found out by chance with Simon that endothelial cells are among the rare cells that can phosphorylate fingolimod. As you know better than I do, fingolimod needs to be phosphorylated in order to be active. And fortunately for us, brain endothelial cells can do that. A monocyte or a lymphocyte cannot do that, but a brain endothelium can do it. So I would suggest that there are probably changes related to the cytoskeletal alterations preceding microparticle release that are targeted by fingolimod. But really we would like to study this in more detail with Simon, but the short answer is that we don't know yet. Sorry.
Okay, thank you. Oh,
Question on basic biology and microvesicles.
Yes?
In a non-inflammatory state, how they remove from the circulation, and does that change?
Well, very interesting question. We know more about their role in pathology than about their role in physiology, except to say that it's been now very clearly established that microparticles, microvesicles, do play an essential role in the triggering of clot formation. They are the actual absolutely number one event that will trigger the formation of the coagulation cascade, etc., because they carry a lot of tissue factor. But apart from that, apart from saying that they play a role in homeostasis, we don't know much about their role in physiology. It's in pathology, and notably in inflammatory pathologies, that we know a little bit more. Now, how are they taken out of the circulation? Again, the short answer is that we don't know, really. They are obviously scavenged by the spleen, by other reticuloendothelial organs, as we used to call them in the 70s or 80s. But again, their role in physiology is much less clear than the role in pathology.
If they express phosphatile theory, be taken up by the spleen in a way
Yes.
similar to nanoparticles or Stephen Miller's red cells with MVP, and that is a tolerogenic process that reduces regulation. Inflammatory state, they're taken up by antigen-presenting cells,
Yes.
and stimulate the immune response.
Absolutely.
So the normal ones are probably going to the spleen and tolerize them. And when you get inflammation, they get taken up. They're very potent. You don't need to put a virus directly into an antigen-presenting cell.
Absolutely.
Antigenic parts quite
Absolutely.
pieces.
Yeah, we've shown that in the field of TB and the field of viral encephalitis. There is a clear role for spreading the antigens by these vesicles. You're absolutely right. Yeah. Absolutely. Thank you.
okay. Thank you, George.
Thank you.
This is the first time I've presented something in the last 5 years that isn't based on my PhD, so it's really refreshing. So today I'm just going to be going over how we're using mass cytometry. This is a collaboration with Simon Hawke and Georges Grau. So I'll give you a bit of a background into why, in particular, we're looking at regulatory B cells. So it's been well established this morning that we have this latitude gradient effect. And it was touched on how there's a lot of evidence suggesting that ultraviolet light exposure is a big contributor to the protective effects of UV. And this is particularly true in animal models. So when we consider how UV can be protective against MS, it's important to realise that it is also capable of suppressing the immune response, in particular the adaptive immune response. So because we're looking at MS, it's this mechanism by which we hypothesize UV may grant protection. So a big question our lab really focuses on is trying to investigate how UV may suppress the immune system. So one of the big things that our lab over the years have found is that UV can activate a subset of regulatory B cells. And last year we published this paper where we found that when we radiate mice with UV and collect their B cells, when those B cells were adoptively transferred into recipient mice, those recipient mice were protected from EAE. So this shows us that the B cells are responsible for this UV-granted protection, or at least it can be transferred by them. And in particular, we believe that it is the regulatory B cells that UV activates. So when it comes to trying to identify regulatory B cells, though, it's quite complicated. So this table here highlights that there are multiple different subsets of B cells that have been found to be regulatory. So this is unlike regulatory T cells, where we have CD25, FOXP3, CD127. At the moment, there aren't any lineage markers to specifically identify regulatory B cells. And furthermore, the same review proposes that any B cell can become regulatory. It's just dependent on the environment in which it finds itself. So this, again, means it's really hard to just identify regulatory B cells based on their phenotype. So to try and overcome this, we adopted the use of mass cytometry. So mass cytometry allows us to look at a much wider range of markers in a single panel when compared to conventional flow cytometry. So for those that aren't familiar with it, essentially we stain the cells with antibodies, much like flow cytometry, but instead they're conjugated to heavy metals rather than fluorochromes. When the cells go through a mass cytometer, everything is incinerated, such that the only things that remain are these heavy metals. The mass cytometer then does time-of-flight mass spectrometry, and that allows us to differentiate between the metals based on their size, and this correlates with what the cells are expressing. So unlike flow cytometry, again, when we have fluorochromes, you get the bleeding or spillover effect. So when fluorochromes are excited, they'll emit some of their own light, and that emission can interfere with the detection of some of the other fluorochromes, and that's where compensation is required. And that's a big limitation at the moment to expand the panels. But with mass cytometry, because we don't have any fluorescence and it's based on mass spectrometry, it means that there's no compensation required and we can have much larger panels. So the panel that we designed, we included a range of different B cell subset markers, some non-B cell markers, mainly for the T cells, some UV-related molecules, some regulatory markers, as well as a bunch of others that we thought would be interesting to investigate. So again, with mass cytometry, it allowed us to look at all of these markers in a single panel. So this is the power that comes with this. So in the context of MS, so based off this panel, we have some PBMCs from just 4 different groups, so healthy, MS untreated, fingolimod or alemtuzumab. This is all still a work in progress and preliminary data, but again, I mainly wanted to show you how we're using mass cytometry in this context. So fingolimod, as we've seen, is an S1P agonist. The main thing here is that it can influence the trafficking of a lot of different leukocytes, whilst alemtuzumab is an anti-CD52 monoclonal antibody. CD52 can be found on a lot of different mature leukocytes. So the big thing here is that both of these treatments, they work, but we still don't, like, they're not very specific. They affect a large range of cells. So we wanted to try and tease apart to see the effect of these drugs were having on different B-cell subsets. And as spoken about earlier, both of these work in very different ways. So not only did we want to try and tease apart the B-cell subsets in MSN untreated, but also to compare between these two different groups to give us a better indication of how these treatments work, but even to try and come up with more specific means of treatment if we can find similar cells that are pathogenic or even beneficial. So I guess a first world problem in this context is that because we have about 35 antibodies, trying to visualize so many markers can be really difficult. So to try and make this easier on ourselves, we use TISNI algorithms. So essentially, this algorithm is capable of grouping cells together based on their level of different marker expression. So of the 35 markers, I selected 9. And then essentially this algorithm can group the cells based on their level of expression of these markers. So again, this is mainly for different B-cell subsets as well as T cells. The other thing I'll mention is that mass cytometry data is exactly the same as flow cytometry data, so you can analyze it in the exact same way. And you can also use these TISNI algorithms for flow cytometry data as well. So these are what the plots look like. Essentially, after the algorithm is done, it just adds 2 extra parameters, so an X and a Y axis. For this plot here, it's just including all 20 samples that we had. And essentially, you can see that there are already just subpopulations of cells. And when we look at heat maps here, so the warmer the color, the more highly expressed it is, we can see that the T cells are all present here. CD4 T cells are here, whilst CD19 and CD20 for the B cells, small population there, and they're present here. Again, this is all just in normal analysis software. We use FlowGel for this. And there are kind of 2 ways that you can go about analyzing this. You can do manual gating on the populations based off the TISNI plot, or you can do conventional gating and then refer back to the TISNI plot. The other thing that we can do here is just look between the 4 different groups, so healthy and MS untreated. So one thing as well to keep in mind, obviously human variability, even though we can't see any striking differences between these 2 groups, human variability will always impact us in that respect. But it does mean that when you do see differences, like with fingolimod and alemtuzumab, you can be sure that this is real. So we can see that the CD3 population here is pretty ablated, not surprising. But what is interesting, especially between these 2 groups, is that we can already see some differences in this B-cell population here. So as I mentioned, you can analyze this much like flow cytometry. So this is just an example, just looking at CD3 positive T cells. So when we gate on them, we can then have a look at where they sit in the TISNI plot and then just overlay it on the original one. And it's exactly where we expected it to be based on the heat map. And then when we're just looking at the T cell frequencies in the healthy MS untreated fingolimod and alemtuzumab, as we saw with the group TISNI plots, we can start to see these differences. But obviously, we're more interested in the B cells. So the way I did the first batch of gating, I gated on CD3 negative, CD27 negative cells. CD27 is expressed on memory B cells. So we're more interested, at least for this example, we're just getting on the non-memory B cells using CD20, CD19. When we have a look at where they lie in the TISNI plot, again, they're where we expected them to be. And this is, again, what we saw with the example of the TISNI plot. We can start to see, when we're looking at the frequency of these B cells of the parent gate, or the frequency of the total cells, we can see, really see big differences between the alemtuzumab and the fingolimod. Possible changes between the healthy and untreated. Again, this is all still preliminary stuff, but based off this, we can really start to tease apart these different B-cell subsets. And we can take this a step further. So based off these B-cells, we can look at the CD38 high, IgM high. So these are transitional B-cells. And again, they're just located in this patch here on the Tisney plots. But this is, again, when we start to see really striking or more striking differences between these 4 different groups. Again, it is still preliminary, but it is pretty consistent, at least the alemtuzumab has much higher levels of these B cells. The other thing to take away, I guess, is that the cause and effect, whether this is an increase in B cells, because this is just frequency, is it just because all other cells have been removed from these patients, or are these being selectively proliferating? That's something we're still going to investigate. But again, just based off this, we can already start to see major differences between these treatment groups. The final aspect I want to show you that we can use this for is trying to give us an indication as to the potential function behind these cells. Because a lot of the markers are included, they're not just for differentiating the different B-cell subsets. We can then have a look at the level of expression of these different markers to see if there are any differences that could give us an indication to the potential function of these cells. So here we're just looking at the median signal intensity. So this is just like the MFI of CD21 and CD25 on these transitional B cells. CD21 is a complement receptor. And it's really striking here that we can see that you've got the healthy B cells, MS untreated. It looks like there's this huge decrease in the level of CD21 expression. It remains low in the fingolimod, but it's increased in the alemtuzumab. So already we can see, again, striking differences between these 2 treatment groups, and this can start to give us an indication as to what these B cells may be capable of doing. But not only that, again, we can really start to see huge differences between just healthy and MS untreated. So that could give us further indication as to, or just more about the disease, maybe even potential biomarkers. CD25, on the other hand, this is interleukin 2 receptor alpha. So regulatory T cells express high levels of this. It can be used by them as a means of suppressing the immune response because it uptakes all localized interleukin 2, preventing the activation of other T cells. So in some contexts, it can be used in an immunoregulatory role. And again, it's interesting here that it's just a huge decrease in the level of CD25 expression on these transitional B cells in MS patients, regardless of treatment. So based off this, it may be that CD25 isn't that important for these B cells, just because, as I said, the fact is both of these treatments work. But again, this is just a really good indication to give us an idea of what these B cells may be capable of. And obviously, due to the interest of time, I'm not going to go into detail for all the other 33 markers, but you kind of get the idea here. So just to summarize, hope I've convinced you that already based off this preliminary data, we can see huge differences between these 4 different groups. Just using this simple panel, I only showed you a few, well, the one subset really of B cells. But really mass cytometry just allows us to really tease apart all these different subsets, as well as give us a strong indication as to their potential function. And the other thing that we're currently working on is developing similar panels to investigate other cell types. So T cells, I showed you, that was the first thing I showed you, huge differences. We can do the exact same thing there. Look at the different T cell subsets, have a look at their level of marker expression to see if that gives us an indication as to their potential function. Innate lymphoid cells has got a lot of interest lately. So essentially, if you don't know, they're just CD45 positive cells. So they're, sorry, having a mental blank. But anyways, they're not T cells, not B cells, not monocytes, not NK cells. nothing, they're just negative for most markers. So there's a lot of interest here because they obviously do something. And of course, we're currently developing a panel as well for monocytes and dendritic cells. So special thanks. Anna helped a lot with the PBMC preparation and the staining. Helen and Tom helped with all the mass cytometry. And my supervisors involved in this are Scott, George, and Simon. Thank you.
Ask a question, Felix. I would imagine your PBMC yield for your big polymer group would be considerably lower, given the nature of the way the drug's working. So I'm assuming that your signalling is adjusted for concentration or for cell count. So is that all being factored into it when you're looking at those differences?
Yep, so as in, when we run them on the mass cytometer, they have to be run at a specific concentration. So that's, yeah, all.
The same CD25 expression in the transition of Tregs of B cells could work the same as Tregs, trapping IL-2 and preventing T cell activation. Could it have the same effect?
So there are groups that have isolated regulatory B cells based on CD25 high expression, but that's not the only marker. As I said, lots of different B cells can be regulatory. There's no defined marker yet. So I think, at least in this context, it gives us an indication that if they are regulatory, they're not using CD25 to be regulatory.
Like Elegant, how you showed a potential mechanism for UV exposure on B cell function. Does UV affect any other cell function?
Yes, it affects a lot. So there are a subset of regulatory T cells that are activated following UV, monocytes, macrophages. Yeah.
Okay, thank you, Felix.